Okay, this is lecture 9, part 3. We're doing the last part of the digestive system, and what you see here is the anatomy drawn of the duodenal or small intestine surface, showing the mucosa, that the mucosa is folded in this kind of manner, and that these folds, of course, increase the total surface area. On top of that, when you look at a magnification anywhere along that surface, you see that there are, if you look in this diagram, you see that there are finger-like projections, which are called villi, and if you look at a magnification, then you see that each villus is, of course, covered by a single cell layer of epithelial cells, and in the center there are blood vessels, as well as a specialized type of lymphatic vessel called a lacteal, which is there to absorb chylomicrons. And if you look at a further magnification here of the epithelial lining, you see that there are individual columnar-shaped cells that have also extensions on their surface, tiny extensions called microvilli. So between the folds, the villi, and the microvilli, there is a huge surface area available for nutrient absorption. When you see a photomicrograph like this, you have to realize that the tissue is in 3 dimensions. It's not going to be perfectly aligned as shown in the drawing. And so when you make a cut, some of the villi will be sliced through, and so you get this appearance of whole villi like this one or this one, but others that look like islands of tissue in the middle of space, empty space, and that's a dead giveaway that you are in fact looking at intestine. One of the major differences in the histology of the small intestine and large intestine is that you see many more mucus secreting goblet cells or mucin secreting goblet cells in the large intestine, and that increase in mucus secretion helps with the movement of compacted feces along the tract. Otherwise, they look fairly similar in histology. Okay, again, diagrams showing the architecture of the tissue in the intestine, the small intestine. You have the layers, the mucosa, the submucosa, and the muscular layer, which below that, which is the muscularis externa. In here, all of this area of the villi, and just underneath the villi, that is mucosa, and then you have submucosa below that. Here you see a photomicrograph showing the villi, and in between the villi there are invaginations, in other words, pockets going down, and these are the intestinal crypts, which are gland-like structures similar to the idea of the gastric pits in the stomach. Here you see a good diagram of the microvilli at the surface of each individual epithelial cell. Large intestine is not really present to, the function is not there to do large-scale absorption of nutrients. By the time the material in the gastrointestinal tract has reached the large intestine, almost all nutrients have been absorbed, and most of the water is being absorbed, but the remaining water is absorbed in the large intestine, and the large intestine will compact the waste into fecal pellets or fecal, I don't know what to call them, sections of feces, I guess, feces. In the large intestine, the remaining water will be absorbed. There are transmembrane channels that allow for absorption or water movement from the space of the intestine into the body tissue, and they are called aquaporins. So these are protein structures that together form a channel for water to be absorbed. Ions also can be absorbed in the large intestine. I should point out that certain infections, bacterial infections, or inflammation can lead to reversal of the flow of water and ions into the gastrointestinal tract, resulting into the large intestine, resulting in diarrhea. Severe diarrhea is actually quite dangerous, particularly in the elderly or small children. Small children particularly will suffer from diarrhea and can actually die quickly. The good thing is that they can also recover fairly quickly. Often when you have an outbreak of diarrhea with poor water quality after some sort of disaster like a flood, earthquake, things like that, they'll often fly in a water treatment, not a water treatment plant, they fly in equipment to produce clean water and fill bags, plastic sterile bags that they've flown in, bags of ion solution, concentrated ion solutions. And they fill it with clean water, and then they give it IV. And the recovery in the children is amazing. They can be brought from near death to stable condition in a very short time. Anyhow, the point here is that diarrhea is due to a reversal of flow of water and ions from the tissues into the large intestine. Okay, now in terms of overall control and regulation of digestion, it can be fairly complicated. There are a lot of regulatory mechanisms operating. Chief among them are neural and hormonal, and we'll discuss quite a bit about the hormonal regulation. Activity in the stomach and the intestine are largely controlled by hormones that are released in an appropriate regulated manner. And some of them, essentially, I've covered, but we're going to cover more here. With arrival of food into the stomach, you get triggering of release of gastrin from cells, endocrine type cells in the mucosa, and they will of course release that gastrin into the bloodstream. All hormones go into the bloodstream. The gastrin will stimulate both parietal cells and chief cells to produce hydrochloric acid and pepsin, and also trigger an increased mobility of the stomach in terms of muscle contraction. The hydrochloric acid, of course, will activate pepsinogen to pepsin. You'll get a lower pH, and that has a negative feedback effect on release of more gastrin. Once the pH drops enough, the gastrin release is inhibited. Meanwhile, the stomach is churning and mixing material and hydrochloric acid and pepsin and turning it into chyme, and chyme will be delivered into the duodenum, the first section of the small intestine, by repeated rhythmic release or relaxation of the pyloric sphincter, the sphincter at the end between the stomach and the duodenum. The arrival of chyme into the duodenum causes a drop in the pH because the chyme is quite acid, and that will trigger the release of secretin, another hormone. So up here we have gastrin, and then down here we have, in the intestinal mucosa, we get release of secretin, which goes into the bloodstream like all hormones, and will have an effect on the pancreas to trigger release of bicarbonate-rich juice or secretions that help neutralize the acid. And the other thing is that arrival of undigested fat and protein within the chyme, in the arrival of that into the intestine, will trigger the release of CCK, or cholecystokinin, and that triggers release of digestive hydrolytic enzymes from the pancreas, and also will trigger release of bile from the gallbladder by triggering contractions of the gallbladder, so that bile salts can go into salts in the bile goes into the intestine and can help emulsify fats. And in that way, the digestive process of the food is enabled, and the food is broken down. Okay. Now, again, let's go through it. This is another diagram essentially showing most of the things that I said before, I would say. Acidic chyme containing fat enters the duodenum from the stomach, triggering release of cholecystokinin and secretin. That's released from the enteroendocrine cells in the wall of the glands in the wall of the intestine. The cholecystokinin and secretin go into the bloodstream and will have an effect on the liver and the gallbladder. Well, let's start with the liver. It will trigger increased production of bile salts, and the secretin will. So an increased production of bile, and also the cholecystokinin that gets released will act on the gallbladder to trigger contraction of the gallbladder and entry of bile into the duodenum. In that way, you have this movement of, let's do a different color, you have this movement of bile salts into this area, and that allows for formation of micelles that can then be absorbed into the mucosal epithelium of the duodenum, and the micelles, that'll cause formation of micelles so that lipase can act on the fat, the emulsified fat, and break it down to monoglycerides and free fatty acids, which will be absorbed into these cells lining the gastric mucosa, and then gets reformed into triglycerides, which form complexes with lipoproteins and go into the bloodstream. The bile salts, I should add, will be reabsorbed into the blood to be transported back to the liver to be used again. There is this enterohepatic recycling of bile salts. Okay. So in terms of other hormones, let's talk a little bit about insulin. Insulin is a hormone produced in a portion of the pancreas. The pancreas is an unusual organ in that it is both endocrine, in other words, forms hormones to be secreted into the bloodstream, and also exocrine, because there's other tissues in the pancreas, exocrine tissue. I'll write that down, endocrine and exocrine. Endocrine tissue produces hormones, and exocrine tissue produces secretions to the outside space of the body. So the exocrine part of the tissue in the pancreas is the part that produces the digestive enzymes that will be exported into the duodenum. Okay, so there's no, I don't know of another example of organ that produces both, has both endocrine and exocrine functions. Maybe the reproductive. Anyhow, let's leave it at this. There are two functions for the pancreas, endocrine. There are two types of tissue there. In terms of endocrine tissue, there are actually two hormones that are produced. We'll talk about the first one, which is insulin. Insulin is produced, and like all hormones, secreted into the blood glucose in response to an elevation in blood plasma glucose levels. When the blood plasma glucose level rises, insulin production release will be from the pancreas will be triggered, and the insulin allows cells throughout the body to, once it binds to its specific receptor on those cells, will allow for glucose uptake by the cells, so the glucose can be used in cellular respiration to generate ATP through aerobic respiration and oxidative phosphorylation. Once that glucose is taken up into the cells, the blood plasma glucose levels drop and insulin secretion slows down and stops. The response to food, nutrients in this case we're talking about carbohydrates in the blood plasma, can be described as in two phases: the absorptive period and the post-absorptive period. During the absorptive period, when blood glucose levels are rising, it's mainly all controlled through insulin. When the blood glucose levels rise, the pancreas secretes insulin. Glucose uptake is increased in most cells in the body. Insulin will also, if there's excess glucose, more than the cells need to take up, the excess will be converted to fat, and insulin will promote fat synthesis and storage of fat in adipose, in other words, fat tissue throughout the body. In a post-absorptive period, after glucose levels have dropped, there's insulin production is downregulated. What you have is a low glucose level, low insulin, and therefore a low insulin level, and so you don't have continuing glucose uptake. The only exception in terms of not being able to take, in terms of the effect of insulin allowing or not allowing glucose uptake, the only exception is in neurons in the nervous system, in the central nervous system. Neurons are able to, in the brain, are able to take up glucose regardless of whether there's insulin or not. They'll continuously need to have a source of energy. If the blood glucose level in overall, however, other than that, however, all the other tissues do need insulin in order to take up glucose. When blood glucose levels fall as a result of the uptake of glucose into cells throughout the body, which was triggered by insulin. When those levels fall, the insulin drops off. If they fall low enough, then another hormone is secreted by the pancreas, and this is called glucagon. Now, what glucagon does is it will stimulate the mobilization of glucose from glycogen stores in the liver. I remind you that the liver is a storage site for glycogen, that up to 10% of the weight of the organ can be glycogen stores, and that glycogen is a polysaccharide made up of many subunits of glucose linked together by glycosidic bonds between the monosaccharide glucose molecules. And with glucagon stimulation, you get mobilization, breakdown of the glycogen, and freeing up of glucose that can then go into the bloodstream to bring the blood glucose level back to the an appropriate level for the body to have enough glucose to function. So let's look at that traffic during the absorptive period. You have food going in. These diagrams show different major tissues that are involved. There are other tissues, of course, but we're going to mainly discuss these: the muscle, fat, liver, pancreas, neural tissue, and gut. And we'll look here in this diagram. Now, this is quite a complicated diagram, and it really shows how many of the metabolic biochemical pathways are interconnected. Some of the things that I'm going to talk about here we've discussed, and others we have not. But it's good for you to be introduced to the fact that there are many interactions and interplays, and that many of the molecular components in terms of nutrient molecules are interconvertible. So if you have amino acids in the gastrointestinal tract, due to the action of protease enzymes on protein, breaking the protein down into individual amino acids, and if you have triglycerides and fatty acids through the action of lipase, and if you have glucose through the action of amylase activity, then that will be absorbed. Remember that all of the nutrients are absorbed in the bloodstream directly or indirectly. The fatty acids and triglycerides will first go into the lymphatic system and then eventually into the blood. In terms of amino acids, all the blood, however, that's draining from the abdominal cavity, the venous return blood, is first going through the liver. In that way, the blood supply that has absorbed nutrients and potentially other types of molecules from the contents of the gut is first being processed through the liver. Amino acids that go to the liver will be in part converted into proteins that are then secreted into the plasma, proteins that are constantly need to be produced and present in the blood plasma, like albumin or clotting factor proteins or complement proteins. So they're circulating proteins. That's where a lot of the amino acids are used by protein production and secretion in the liver. Amino acids will also go to other tissues. One of the major destinations would be muscle, which is almost all protein after all. The other kind of molecule that is taken up are triglycerides and fatty acids. They go into the lymphatics and then into the blood, and the muscle can take that up. Under certain circumstances, muscle will use fat as a source of glucose. It can convert the fat to by gluconeogenesis into glucose and use that as for energy. Triglycerides also will be taken up in the liver and processed there and transported via and covered with VLDL and transported to other organs. Also LDLs will circulate that. LDLs will bring some of the triglycerides back. LDLs are considered bad. It's considered bad to have too high a level of LDL because it can bring cholesterol back into the circulation. Triglycerides also will go and get stored away in adipose tissue if you're eating too much of too fatty a diet, and they can either go well, I don't know why there's two arrows. It can either go directly or first be processed through the liver. In terms of glucose, there's always glucose uptake by neural tissue. The brain always needs a steady source of glucose. Other organs would require insulin release and interaction with the insulin receptors before they can take it up. The increase in glucose in the blood plasma associated with eating carbohydrates does trigger insulin release from the pancreas, and that will act to allow many tissues to take up glucose, but particularly in the liver, if there's excess glucose, it will be taken up and stored as glycogen. It can also be stored away as fat in the liver, but mostly it's stored away as a source of as glycogen. Some of it is used for energy in the liver, but for the most part it's stored away. If there's excess, it's stored away as glycogen, although some can be used certainly to make be converted to fat. What else is there? Insulin will also increase the activity of adipose tissue to take up glucose and store it away as triglyceride if you have excess amounts of glucose in your diet. Okay. It can also trigger uptake of glucose by muscle. Glucose, of course, in muscle will be converted to glycogen and stored there and used there as an energy, as a supply of molecules that can be oxidized to produce ATP. In the post-absorptive period, you have different kinds of trafficking of molecules. Here, remember that the blood glucose level has dropped, and the person is not hasn't been eating, for a little while at least, and you get a different kind of dynamic. A low blood glucose level will, of course, trigger the pancreas to produce glucagon, and glucagon will act on the liver to mobilize glucose from glycogen stores, and glucose will then go into the bloodstream to supply other organs. The neural tissue always needs some glucose, but all the other cells also need it, certainly in order to generate energy. However, without insulin, those tissues cannot get it, but once the blood glucose level rises enough, the pancreas will produce start producing insulin so that they can take it up. This is kind of the background continuous, enough in glucose to have for neural tissue, because of the constant need in the CNS, central nervous system. Meanwhile, glucagon will upregulate, or in other words, increase, because even if you don't eat glucose, you still need it, and the body will first mobilize glucose. If you don't have glucose in you, in your gastrointestinal tract, if your blood plasma glucose levels have been dropping, then if they drop far enough, then the release of glucagon will also trigger a process called gluconeogenesis, the production of glucose from non-carbohydrate sources. And that's what you see here. that amino acids can be used for production of glucose through gluconeogenesis. So protein can be a source of molecules for glucose to be synthesized, and this process is done in the liver. It's done in also kidneys, but we're not going to get into that. Mostly it's done in the liver. So that's gluconeogenesis through using protein, amino acids, but also fat can also contribute to that. We'll see that soon enough. So that's a way of making glucose. Glucose can also be, I'm not sure why oh, here it is. The liver also contains some fat in the liver, and that triglyceride or fat can be converted, will be complex with VLDL, and that can lead to LDLs bringing it back or bringing the fat to muscle, and fat can be used in the muscle for energy. Triglycerides are also broken down and release fatty acids, and the free fatty acids can be used for energy in the muscle. Production of ATP. Here you see protein releasing the amino acids to make available amino acids for the liver to produce gluconeogenesis. I think, you know, all these interactions are perhaps beyond the level of this course, but I think it's worthwhile for you to be exposed to these pathways and understand that there are many, many interactions, that many of these key nutrient molecules can be converted one to the other, so that we have these reserve fallback regulatory mechanisms operating, particularly when you don't have free available nutrients coming from the gastrointestinal tract, and you're in the in other words, you're in a post-absorptive period. Okay, in terms of triglycerides from the adipose tissue, release of that glucagon presence will increase mobilization of free fatty acid from the adipose tissue, and releasing it so that it can undergo a metabolic process that allows for ATP production. I'll make it simpler and generalized. Okay, let's move on. All of those interactions and pathways, what I haven't previously covered, what I just spoke about on that slide, it's more for your own information. I would not necessarily at all expect you to know it on an exam. Okay, well, this is the same idea, regulation or homeostatic maintenance of appropriate levels of glucose in the blood operating through insulin. When there's glucose, insulin gets released and allowing it to be used, glucose to be used by cells, allowing for storage away as excess as glycogen in the liver or as fat in adipose tissue. With uptake of the glucose level will fall. If it falls far enough, you get glucagon being produced, and glucagon will mobilize glucose from glycogen stores in the liver so that it'll go into the bloodstream and you'll have enough for other cells. Also, fat storage cells are acted on, triggered by glucagon to start breaking down triglycerides and freeing fatty acids, releasing them into the bloodstream, and they can be used through a process which happens to be called beta oxidation. It will generate ATP in neural tissue and other tissues. Okay, this is the same idea about increasing, blood glucose leading to insulin secretion, decreasing blood glucose leading to glucagon, and always a balance. The blood glucose level, the body tries to maintain it at within a certain defined range so that it's available in adequate quantities for energy production. There's nothing that I haven't spoken about before here. A lot of that slide that had all those interactions, pre-absorptive, post-absorptive, or during the absorptive period and post-absorptive period, a lot of these things I am now mentioning, like the fact that glucagon will mobilize glucose from glycogen by allowing for breakdown of glycogen in the liver, freeing of glucose to the bloodstream, and that it causes breakdown of triglycerides to release fatty acids that will lead to, some of it can get converted by gluconeogenesis to glucose. Now, in terms of abnormalities, people who have a disease called diabetes mellitus can end up experiencing abnormally high pathological levels of glucose in their blood. That's a problem. The reason the blood glucose levels go so high, that the concentration of glucose in the plasma is too high, is because they either do not synthesize adequate levels of insulin in response, and therefore most cells in the body cannot take up the glucose, because in order to take up glucose they need insulin interacting with the insulin receptors on their surface to permit them to take up the glucose, and so that maintains the high glucose level in their bloodstream. It's not being taken up by cells, and therefore it remains high. And that type of inadequate insulin response is called type 1 diabetes. There are also some individuals, and this is usually with occurring later in life, mature onset, or type 2 diabetes, in which the receptor is defective. Chronic glucose imbalance can be associated with a wide range of serious outcomes, including blindness, heart failure, and reduction of circulation in the lungs, leading to loss of tissue in the legs, toes, feet, etc. Okay, type 2 diabetes, also called mature onset, also called adult onset, also called non-insulin dependent diabetes. Some individuals have genetic predisposition for development of type 2 diabetes. There's strong evidence that it's associated with high nutrient uptake and lower physical activity. These are key risk factors associated with developing type 2 diabetes. Too rich a diet, and too little physical action. The genetic predisposition to this kind of mature onset of disease is particularly clear in certain populations. This is old data, but nevertheless fairly instructive, of natives in, I believe, southwestern United States. Percent of individuals who develop type 2 diabetes by age, and this shows a progressive increase as the income in the population increased, and people had a more and more rich diet and were less active, and so you see increasing levels with increasing time. I mean, these are astonishingly high percentages of the people, greater than 70% of the adults with type 2 diabetes. It's serious. Here is association with obesity, and this data is for females, looking at increasing obesity and age, and again, very high levels. extremely high levels, quite a serious disease process. Yes, these people have a genetic predisposition, but these are maybe the extreme and higher levels, but the same applies to other individuals in our population. If you gain enough weight and remain inactive, you have a pretty high risk of developing this kind of disease process. And so that's the end of this lecture 9, part 3, on the digestive system and nutrition and regulation of nutrient balances.